So thanks a lot to the organizers. It's been a great meeting so far. I'm looking forward to continuing to chat with everyone. I'm going to talk today about sort of temporal processing from a perspective that I began thinking about it from more and more, and that is the perspective of control. Because for me, at the end of the day, the brain is for control. It's for controlling the behavior and the physiology of organisms in complex and dynamic environments. And 600 million years of nervous system evolution have endowed nervous systems with a range of different mechanisms for producing that control. For instance, we have mechanisms in the brainstem and spinal cord that we think of as being responsible for more automatized forms of control, reflexes, central pattern generators, movement primitives. And of course, these kinds of mechanisms can be selected, sequenced, or otherwise modulated by descending input from a number of different brain systems that are positioned more anterior along the neuraxis that we think of as depending more centrally on their capacity to learn through experience within the lifetime of an organism. And so, all of these different circuits for control depend critically on temporal regularities in the environment or in the subset of the environment that they have to control. And some of those temporal regularities are those that have existed and are stable on evolutionary timescales, and those sort of seem to get embedded in relatively hardwired or innate circuitry, innate mechanisms for control, whereas other ones are more idiosyncratic from the perspective of a species and are specific to the individual environment or individual experiences of an individual of a species. And so, for those kinds of temporal regularities, we have a variety of different learning systems. Now, each of these learning systems, they possess sort of stereotyped circuit architecture that suggests that they make some kind of core computational contribution to the functioning of the whole. And so I'm going to talk about, one of these systems, the basal ganglia today, but I think it's important to keep in mind that each of these systems, is processing temporal information and presumably for some distinct kind of contribution to the overall kind of distributed control system that is the brain. So this different sort of modular architecture of the nervous system we see is what I'm referring to as the heterarchy of neural mechanisms for control, because there's distinction between these different systems, but there's not an obvious ordering between them. This term heterarchy I first read in the writings of Warren McCulloch, and the point is that even mechanisms that we think of as being relatively low level, like in the spinal cord, like a reflex, they can take complete control over behavior, So they're not in any way, in any stable way, sort of slaves to these systems that we think of as being higher up in the nervous system. So there's a difference amongst control mechanisms without there being an obvious ordering. However, the brain is obviously operating on information that's arranged sort of hierarchically in terms of sort of degree of abstraction from the immediate sensory motor state of the animal. There are different transformations that go on as you move deeper and deeper into the brain. And all of these learning systems, in particular, they seem to operate on many different levels of information that are organized hierarchically. So I'm going to touch on these sort of two dimensions, which I think of as fundamental for neural mechanisms for behavioral control, and sort of look at them through the lens of temporal processing today. Now, as I mentioned, I'm going to focus on one of these brain systems, the basal ganglia, today. I will branch out into talking about some other brain systems because I'm very interested, and have been for a long time, in control mechanisms that involve learning. And the basal ganglia has been implicated as being particularly important in learned kind of behavioral programs for control. And those ideas are very much inspired by a field that grew out of sort of cybernetics and animal learning theory called reinforcement learning. And the first thing I want to point out is that reinforcement learning is not an algorithm. okay? It's a behavioral problem, all right? And the problem is defined, as was said here by Rich Sutton and Andy Bardo in their sort of Bible of reinforcement learning. Reinforcement learning is defined not by characterizing learning algorithms, but by characterizing a learning problem. And that problem is how do agents, or animals in this case, learn how to behave through interaction with the environment, okay? And this is also a kind of form of an optimal control theory where the objective of a behavioral control is to maximize cumulative reward, okay? So reinforcement learning is a problem. It's not an algorithm, but the field is constantly generating a kind of expanding set of algorithms. And here's just a sample from this tutorial page from OpenAI showing a kind of tree of different reinforcement learning algorithms that exist today, from which we as neuroscientists, I think, can draw inspiration for thinking about how particular kind of components of the nervous system are contributing to behavioral control. The other thing I want to point out is that even though these all might be referred to as reinforcement learning algorithms, in most cases they involve actually a number of different kinds of learning, okay? So for instance, let's take this one right here, deep Q networks, okay? This is a deep network that does representation learning in order to develop efficient and useful representations of the environment. And then from that representation learning process, you're also learning policies for behavioral control, okay? So when we talk about reinforcement learning, I just want to be very clear, I'm talking about the whole enchilada, this sort of collection of different computations that are useful for producing behavioral control. And I'm not talking about the thing that people often focus on in neuroscience, which is just like learning about reward, okay? Okay, so as I mentioned, you know, people have now long associated the basal ganglia with reinforcement learning, and that's because of this view that's developed over the past 30 years that sort of holds the basal ganglia as what in the parlance of reinforcement learning is called a policy learning system. So all reinforcement learning algorithms, they essentially have as their goal to learn these programs for behavioral control, and these programs are called policies. And the dominant view that's evolved in terms of the contribution of the basal ganglia to this kind of process goes as follows. So this is a lateral view, the schematic of a mouse brain, where I've highlighted this collection of subcortical nuclei here in red. Collectively, we refer to as the basal ganglia. And the idea is that input from an extremely wide variety of territories in cortex and also the thalamus provide the basal ganglia with information about the sort of current state or situation that the animal finds itself in, and the actions that are on offer in that situation. And the state and action-related information converges in the major input area of the basal ganglia, the striatum, with extremely dense input from midbrain dopamine neurons that are known to carry what looks like an important teaching signal from computational models of reinforcement learning called a reward prediction error. And it's the convergence of this teaching signal with the state and action-related information that's thought to produce plasticity at striatal synapses that allows the basal ganglia to learn something about the value of the actions that are available in the current state of the world. And this action value-related information can be used to derive policies for behavioral control, which can then influence the selection of action through the various extrinsic connections of the basal ganglia with downstream motor structures either in the brainstem or indirectly in cortex through thalamocortical circuits. Okay? So this is sort of a very general view here. And today I'm going to talk about how this sort of general view applies to a particular set of behaviors that involve animals, rats and mice, making use of temporal information to either make judgments about duration or to make predictions about rewards in time. Okay? And in the first story I'm going to tell you about really is useful, I think, for highlighting an example of this idea of heterarchy between different control functions for different brain systems. Because when we looked into the basal ganglia, during this task, what we found, and started to perturb the basal ganglia during this task, what we found is that it didn't seem to be responsible for certain aspects of behavior, which then suggested maybe a certain division of labor between brain systems. We've since moved out to start to ask questions about other brain systems. Okay, so in this first part of the talk, I'm going to focus on a two alternative forced choice interval discrimination task. In the second part, I'm going to focus on a simple classical conditioning task in order to ask questions about how the reward system learns to predict information about the distribution of rewards in time. And this turns out that it's enabled by a certain sort of multi-scale property of temporal discounting within the dopaminergic system. And that sort of multi-scale temporal discounting property, I think, is a nice example of sort of a hierarchy or hierarchical processing with respect to time. Okay, so the task I'm going to talk about in the first part is a simple two alternative forced choice task. Animals are placed in a behavioral box. There are 3 nose ports positioned along a wall. And they learn to initiate trials by inserting their snout into a central port. This elicits a brief auditory tone, followed by a variable time delay before a second tone is played. And at this point, animals are free to move to the left or the right port to get reward, depending on whether they believe the time delay between those 2 tones was shorter or longer than a learned category boundary. Okay? So animals are making categorical long-short judgments about the interval of silence between 2 tones. And I just want to point out here that at the time of the second tone, there's nothing in the environment that tells the animal where to go to get reward. Okay? Instead, they have to rely on this sort of latent, dynamic, evolving internal representation related to elapsed time. And I think this sort of flexible operation on dynamic, latent internal variables is a real hallmark of many processes that we would refer to as cognitive. So in addition to, I think, providing us an opportunity to ask questions about the representation of temporal information, I think there are some maybe general lessons to be learned by studying a task like this about cognition. Okay, so animals can learn to perform this task quite well. Here I'm showing a representation of their behavior with the probability that they make a long choice as a function of the intervals that we present to them. And you can see that animals perform almost perfectly when the intervals are easy, far from the decision boundary, but their choices become more variable as the intervals approach the decision boundary. Okay? And so this variability in animals' judgments is a lever that we can then use to interrogate neural systems, because we can ask where in the brain do we find neural activity that co-varies with that variability in choice, all else being equal, and where, when we perturb neural circuits, do we induce systematic changes in that judgment. Okay, so taking that sort of general view of the basal ganglia as a policy learning system that's sort of mapping together states and actions in order to compose these sort of programs for behavioral control. In this task, what we're asking the animal to do is to sort of map state variables related to elapsed time onto one of 2 actions, to go to the left or the right. Okay? And so in order to do that, we assumed it would need some sort of temporal basis, right, for mapping together sort of time information and action. And so in a series of experiments, we recorded activity from dorsal striatum during this task. And what we found in a variety of tasks is sort of depicted here. And I think Dean showed actually this figure in his talk. And that is that different neurons in the striatum are active at different times during the delay period when we're asking animals to keep track of this interval duration. Okay, so what that means is that at a population level, the state of the population is continuously changing and it's not repeating itself during the interval, and that's precisely what you need in a system that you might use to kind of encode elapsed time, right? However, if the animal is actually using this pattern of activity to guide its choices, then when this pattern evolves more quickly or slowly, okay, there should be some systematic differences in the animal's judgments. Okay, so to look at that, we can sort of recast this pattern of activity in population state space, which you just heard Ugo talk about. as a trajectory. So when the first tone is played, the state of the population might be here, and then it evolves along a reproducible trajectory in the state space defined by the activity of all the neurons in this population. And then if we are to look at simultaneously recorded populations of cells, and this is just between 10 and 20 some odd cells at a time, we can characterize the state of the population at the offset of one of the intervals, say this near boundary short interval here, which might correspond to this schematized point here. And then we can project that point onto the typical trajectory. Okay? When we do that, what that gives us is a sort of number that quantifies how fast on average the population has evolved along that trajectory up until that point in time when we're asking the animal to make a judgment. Right? Now, of course, from trial to trial, that mapping is variable, and then we can go and exploit that variability and ask whether it relates to animals' judgments. So for instance, if we take the average speed here, the middle of this distribution of mappings, and look at the probability that the animal makes a long choice, we might see something like this. We take the faster third, we might see this, slower third, this. And if we do this across all the different intervals in our interval set, we see there's this systematic relationship between the speed metric, which we extracted from simultaneously recorded cells in single trials, and the animal's apparent kind of speed of their timekeeping that they're using to make these judgments. Okay, so this is a nice correlation, and we found many others, and have done lots of other analyses to show that there's a really nice tight correlation between population activity in the striatum, and particularly the speed kind of metric that you can extract from the population in the striatum and animals' judgments. However, of course, correlation does not imply causation. And so what we'd really like to do to test the hypothesis that this speed metric is really responsible for guiding the speed of their decision process that they're using to make their temporal judgments is to take experimental control over this feature of the population response, right? And just sort of leave the general structure intact, but speed it up and slow it down, right? Turns out that's a kind of hard thing to do, even with very fancy kind of optogenetic tools. So we turn to a relatively lower tech approach, which is to use temperature. And that's because in a variety of different systems, temperature has been shown to be capable of changing the sort of temporal scaling of patterns of population activity while leaving the structure intact. Okay, and this is just an example from an invertebrate system, the crab stomatogastric ganglion, Eden Martyr's lab. You can see the activity of these 3 neurons in this central pattern generator circuit, which guides the pyloric rhythm in the crab at 7 degrees Celsius, 11 or 19, and you can see the frequency of these oscillations just increase with temperature, but if you normalize by phase, all of the phase relationships are constant. Okay, so the structure and population activity is the same, you've just changed the temporal scaling. And of course, we've seen temperature used to rescale aspects of behavior. So this is data from Michael Long when he was in Michael Fee's lab, where they set out to test the hypothesis that area HVC in the birdsong circuit which has very precise firing at particular phases of the bird's song, was responsible for actually setting the timing of the bird's song. And indeed, when they warmed or cooled area HVC using the solid state heat pump, they found that they could contract and stretch the bird's song while leaving other aspects of the structure and the spectral features intact. So we, together with the hardware and software platform and the Champalimaud Foundation, developed our own thermoelectric device for focal closed loop control of temperature, in this case in the striatum. You can see a schematic of that here. So we have this Peltier solid state heat pump coupled to 2 silver wires which are insulated down to their tips, and then those tips are implanted bilaterally in the striatum. And the first thing we wanted to do is to sort of test what is it that temperature does to striatal population dynamics. Every single element of the physical world has some temperature dependence, so it's not obvious that all of the differing sort of temporal dependencies of the components that make up neural tissue and neural circuits, that by changing the temperature, you're in every case going to simply rescale population activity. So we wanted to check and see what doing this in the striatum actually did. And to do this, we wanted very reliable reproducible patterns of population activity. And so we adopted a paradigm that was developed by one of Hugo's colleagues, Pavel Rueda-Orozco at UNAM in Euraquía, wherein an anesthetized animal under urethane anesthesia has channelrhodopsin expressed in somatosensory thalamic structure, ventrobasal complex. We then insert an optic fiber over the thalamus, a neuropixels probe into the striatum, and then our temperature probe. And then we can stimulate relatively long-lasting bouts of population dynamics by stimulating these channelrhodopsin expressing cells, and then subject those populations to differing temperatures and ask what does temperature do to population activity. Okay, so I'm going to show you a couple different example neurons from this paradigm. With time on the x-axis aligned on the optogenetic stimulation, and then trial number, or really pulse number, on the y-axis. You can see this schematic here showing how we're manipulating temperature in blocks of 3 minutes. So we're always going from a control temperature to a cool, control, colder, control, warm, so on and so forth. So 2 different cool temperatures, 1 warm, always with a control temperature near body temperature in between. You can see initially there's a direct response to the stimulation, but then in the sort of slower relaxation dynamics of the population, there's this later phase of the response that we're able to just push around, rescaling in time using temperature. Right? So if you just look at the spike density function above, for this neuron, it looks like this. This is another neuron. And if we look at the population, you see there's a very clear, systematic, monotonic effect of temperature on the temporal scaling of responses. Okay, so this is now expressed as the percent dilation of the spike density function relative to a subset of the control childs. Okay, so you just take the control spike density function, you rescale it by a lot of different factors, and you ask when does it best correlate with that particular temperature's spike density function. Okay, so great. It looks like we have a tool with which we can manipulate this particular feature of the population. However, temperature didn't only affect temporal scaling. It actually had a qualitatively distinct effect on another aspect of neural activity, and that was just baseline firing rate. And this turns out to be a peculiarity of striatal circuits, because striatal neurons express a particular potassium channel, which passes more current at warmer temperatures. So both warming and cooling lowers baseline excitability of neurons. Right? And this is going to end up being important in a few moments. So I just want you to put a pin in this. Right? Nonetheless, we do have a tool with which we can affect this kind of rescaling in time. So what happens when we do this in the context of a behaving animal? So this is now the animal's choice behavior in the control temperature block. Actually, this is aggregate data from 6 animals. The warm temperature block, cool, cooler. So indeed, systematic bidirectional dose-dependent effect of temperature on the animal's psychometric curve. So when you cool down the striatum, animals are more likely to underestimate duration. When you heat it up, they're more likely to overestimate. This is aggregated across 6 animals. This effect was really robust. So this is now data for each individual animal in this cohort, now expressed as the percent dilation of the threshold parameter of these fit psychometric functions. Okay, every single animal, bidirectional dose-dependent effect of temperature on their choices. So we're done, right? We showed there's this nice correlation between population scaling. Now we've gone on into the perturbation, and we've found that indeed these dynamics seem to have a causal role in controlling the temporal evolution of the animal's decision. However, when we were thinking about these experiments, we thought, there's the basal ganglia is implicated in motor control, and there's evidence from the behavioral literature that animals sort of develop stereotyped behavioral patterns. Okay, not sure why that happened. Where'd it go? they often develop stereotype behavioral patterns during timing tasks, which has led some people to speculate that maybe organisms are sort of offloading timing function onto motor circuits that have evolved to produce, very precise time-varying patterns of activity. So we thought maybe if we saw these kinds of effects, we'll also see similar kinds of temporal scaling of just moment-by-moment behavior. Right? That's not what we observed. So first, temperature didn't similarly affect the path that animals took when they executed their choice movements. So if we just track the animal's head as they're moving to the different choice ports, okay, here are all the individual track paths from individual trials. Here are the averages for the different temperatures. If we splay that out in time, okay, no obvious differences in the position of the animal over time. And the sort of overall envelope of velocity around these choice movements doesn't seem significantly different. However, there was a modest effect of temperature on the speed, the average speed with which animals executed their choice movements. But it was qualitatively distinct from the effect on choices, okay? Both warming and cooling slowed down the animal's choice movements, okay? And of course, this resembles much more the effect that we observed of temperature on just overall activity of striatal neurons, okay? So, how do we interpret this? Well, if you think about these different measures of behavior I'm showing over here, these are sort of aspects of the kind of continuous moment-by-moment control of behavior, right? Whereas, when we are training the animals to do this task, what we're doing is we're actually asking them to place kind of a discrete transition between making a movement to two different options, okay? And so we wondered whether what all of this data was telling us was that in general, the basal ganglia seems to be kind of preferentially involved in what you might call kind of discrete forms of control, where the space of actions are relatively compact, discretized, and its sort of lone contribution to kind of continuous motor control is through a kind of low-dimensional gain signal that we associate with, say, vigor, which is something that basal ganglia has long been implicated in, and that we know is related to things like dopaminergic tone in the basal ganglia, and indeed, our data from our lab and other labs has shown you can simply impose a very low-dimensional manipulation of striatal circuits, say, just inhibiting the direct pathway in sensory motor striatum, and you can just slow down animals' movements without changing other features of those movements, okay? So this has led us to start to speculate a little bit about, again, this division of labor between different brain systems that I mentioned at the beginning of the talk. And I just want to kind of outline that speculative thinking for you right now. So as I said, kind of all reinforcement learning algorithms, they involve learning a policy or a program for behavioral control that maps states of the world to actions under this subjective and maximizing reward. And the kinds of reinforcement learning algorithms that are associated in neuroscience with the basal ganglia are so-called value-based reinforcement learning algorithms. Okay, and what this means is that the policies are derived from intermediary representations of the value of actions. Okay, and the way that learning proceeds in these kind of value-based reinforcement learning algorithms might go something as follows. So, say you come into some state of the world, S of t, and you're considering these 6 different actions. Okay, here, and you have some estimate of the value of those actions here encoded as this Q value or the quality of actions. Okay, and let's say this is the set of values you observe in your system, so you might choose action 4 because it's the most valuable. Now let's say the outcome is worse than you expected. Well, then we expect that there's this negative error signal which drives down the value of action 4, and so the next time you come into this state, you might choose action 6. Now, if you're only considering a space of 6 actions, this kind of jumping around as you're trying to converge on the right policy for this state is okay. You will still relatively quickly converge on a good policy for that state, that context. However, if instead your space of actions is very high dimensional and continuous, okay, this kind of algorithm, this kind of jumping around is disastrous, okay? You won't converge. It'll be incredibly sample inefficient. Engineers who have to deal with the variety of kind of discrete and continuous control problems that, say, present themselves to robotic systems are well aware of the fact that you're better off using different algorithms for these different classes of control problems. Now, value-based reinforcement learning algorithms are not the only reinforcement learning algorithms out there, as I showed at the beginning of the talk. And the basal ganglia is not the only kind of learning system involved in motor control out there. In fact, it's not even the one that we would most often associate with continuous control and coordination. We heard from Megan yesterday about another brain system, right, which is seated back here in the hindbrain, which we much more often associate with problems related to continuous control. And so, though historically, many of the computational theories have kind of proposed for how the cerebellum contributes to motor control, have sort of proposed, for instance, that it might implement supervised learning algorithms for learning things like forward models for control. I'm gonna speculate a bit and say that we can actually view in an entirely consistent way with those old theories the cerebellum as another reinforcement learning system. Okay, and actually recently people have started to observe more and more reward-related signaling in the cerebellum, which has gotten people thinking about cerebellum and reinforcement learning. And perhaps it's involved in implementing a different class of algorithms that are better suited for continuous control. And those are algorithms where your policy is learned directly. So you don't go through these intermediary representations of value. Instead, what you do is you parameterize this high-dimensional continuous space of actions using some set of parameters, okay? And this defines some space. And the way learning proceeds when you learn your policy directly is to try and, is through a kind of gradient ascent or gradient descent method. And so learning is smooth. You're trying to kind of climb this landscape of expected reward. And so if you're near the region of your optimal policy, you will converge to it smoothly. You won't jump around the way you do with these value-based types of algorithms. And in fact, the best algorithms for the kind of hybrid control demands that, say, controlling a robot presents, the best ones are ones that combine the kind of best of these two worlds. And you can kind of see why, right? Here, these kinds of algorithms allow you to converge smoothly to an optimal policy when you're in a sort of continuous action space. These kinds of algorithms allow you to jump around. So for example, you can avoid the problems associated with gradient methods if you don't have a fully convex problem. Okay, so that's speculation, but we've recently started to kind of step out. What am I doing on time? 33? Perfect. Okay, so to step out and study other behaviors that more explicitly require kind of continuous control and coordination. And so we've begun focusing on this head-restrained four-limb reaching task in mice. And we've started developing this task in collaboration with John Krakauer and Megan Carey. And basically, you can see the basic version of the paradigm, which involves the animal sits paws on a perch, and then it learns to reach out and grab this water droplet and bring it to their mouth. We can characterize the movement at very high temporal resolution, taking high-speed video at 400 hertz. And then we've started by just applying these same kinds of temperature manipulations now to cerebellar cortex. Okay, this data is very preliminary, so take it with a grain of salt. But what we're finding is sort of consistent with that general view that I presented to you in the previous slide. So if we look at these keyframes from these different trials of this task, where we've cooled, warmed, or held the cerebellar cortex at a control temperature, these keyframes are taken from the point of maximum extent of the reach, and you can already see one effect that we seem to observe of temperature, and that is that when you cool down the cerebellum, animals' reaches become hypermetric, so they overreach, right? And when you warm up, they're hypometric, so they underreach. Okay? Now if we look at the video, you can kind of see what's going on. This is a control trial. This is a warm trial. Okay? And this is a cool trial. So they actually miss. And if you think about when we heat up the cerebellum, and if you think about the kind of moment by moment commands required for continuous control that would compose a kind of learned continuous control policy, sort of existing in different time bins, and then you contracted that time base. Okay, what you would expect to actually see is that the whole kind of movement trajectory becomes contracted, right, and less eccentric. It's because you're going to start turning earlier, right? And conversely, when you cool things down. So those are just some example trials. This is just some aggregate data from an individual animal, plotting the X, Y, and Z position of the forepaw on cooled and warm trials. And another measure you can look at is just to look at the autocorrelation of the paw movements. Okay, and when you do that in the cerebellum, you see that there's this systematic relationship in the sort of temporal scaling of the autocorrelation when you cool the cerebellum that we don't see when you do the same manipulation in the same task in the striatum. Okay, so in that first part of the talk, I talked about sort of how trying to understand temporal processing in real time and sort of how it guides the placement of different actions at different times in the context of a decision task relates to population dynamics in the striatum, and how perturbations of the speed or the temporal scaling of those dynamics revealed a sort of surprising potential distribution of labor between, say, the basal ganglia and another learning system for motor control. And then we're currently following that up. In the second part, I'm going to switch gears a little bit and talk about a problem that's more related to how neural circuits learn to predict information about the future time of relevant events in the environment, particularly rewards. Okay? This involves a different behavior, and in order to kind of get into the details of these results, I need to kind of back up a little bit and describe a little bit about temporal difference learning. So how many people here kind of heard of temporal difference learning, understand how it works, could write down the temporal difference RPE equation from memory? No? Okay, so so I think it's a good idea to just sort of go through a little bit the principles behind this particular class of reinforcement learning algorithms called temporal difference learning algorithms that have been really influential in neuroscience. I'm going to go through this in a very simple kind of behavioral context, which is classical conditioning, wherein we want to train this model to learn whether a cue predicts reward. Okay? And the first thing you have to do with these algorithms, you have to represent the stimulus over time, okay? Which in this formulation, we're going to do using this sort of set of basis functions, x0 through x3. Okay? And they just represent the fact that the conditioned stimulus has occurred at various different time points into the past. Okay? So that's the first thing you have to do. And I would just point out, you might see some resemblance between this very artificial kind of representation of time since a cue was delivered and this kind of pattern of activity that I showed you in the striatum. Second thing you have to do is you have to learn a so-called value function, okay? And this is a function that encodes the expectation of cumulative future rewards, right? And the way we're going to do that in this formulation of the problem is by just summing up a weighted combination of these different basis functions, okay? So that's our value function. How do you learn those weights? Well, you have to compute this thing called the reward prediction error, which is essentially a signed surprise signal, right? And this is why this algorithm is called temporal difference, because the prediction error is computed as the temporal difference between the value function, the temporally discounted value function at time t plus 1 minus the value function at time t, it's the temporal difference part, plus whatever reward is coming in at time t. All right? That sign surprise signal is then used to update these weights. Okay? And so you just kind of run this series of steps as your agent experiences this very simple world. And we'll show you sort of step by step how information about sort of predicted future reward is learned. Okay? So let's say we come into this very simple world. We can make this a little bit more neural. We can assume that each of these different functions actually represent the activity of an individual neuron whose outputs are pooling onto a downstream unit that's supposed to learn this value function. Right? So we come into this world. We don't have any weights here. We don't have any predictions. And so on trial 1, our value function is 0 at all time points. Right? And we come along here. We're computing our prediction error. Value is 0 everywhere, so this is 0 everywhere. But then when we get to this reward, okay, now this prediction error has a non-zero value. So on trial 1, before I've learned anything, when I see the reward, I get a prediction error at the time of reward. Right? So that's before learning. Now, because I had this prediction error here, right, when I go to update my weights, I see, oh, look, I'm going to multiply my prediction error times my set of basis functions. The only one that has a non-zero value is W3. So I'm going to add a little bit of weight to the connection between this neuron and my value function. Such that on trial 2, when I come in, now I have a little bit of a value prediction at this point in time. Okay? So then, as I sort of march along in time here, okay, value function is 0 at the beginning here. This term is still 0, no reward yet. But when I get to this point, okay, now this difference is positive, right? Because my value function at time t plus 1 is greater than my value function at time t. So I have a little positive prediction error now here, right? However, when I see the reward, now my temporal difference here is negative, but it's canceled out by the fact that there's a reward coming in. So I just have a little prediction error here right before I had it on the first trial, okay? But I no longer have a prediction error at the time of reward, right? So now I go again, I add a little bit of weight here now to my connection between x2 and my value function. All right, so on trial 3, my value prediction has moved back a little bit, right? And so the way this algorithm learns is by sort of back propagating this information about reward until the earliest point in time when the system has some sort of predictive knowledge of that future reward, right? So after learning, now instead of having a prediction error at the time of reward, you now have it at the time of the predictive cue. Okay, so what happens when we emit the reward? Okay, after we've learned. Well, now I come in here. I still have my prediction error here at the time of the cue. When I get to this point, right, this term is negative, but now I no longer have a reward to cancel that out, right? So I actually have a negative prediction error at the time of the expected reward, okay? At the right time, I should point out here, because this system has access to information about elapsed time. And so this kind of profile in this very simple kind of impoverished world where the system just has to learn that a cue predicts reward, of having a prediction error at the time of reward before you've learned anything, a prediction error at the time of the cue after you've learned something, and then this dip when you emit a predicted reward, is what has really driven an explosion of research in reinforcement learning in neuroscience, because it was observed that the phasic activity of these midbrain dopamine neurons bears striking resemblance to this sort of pattern of responses that I just showed you in this model. However, there's a problem, okay? Which is that because the way this algorithm works is it's sort of combining information about reward delay and reward magnitude into a single scalar, okay? You're losing information about when the reward is going to come, right? And you can see that by just considering that actually when you take into account this discounting term right here, okay, your value function, right, as a function of time aligned on delivery of Q is going to look like this sort of ramping signal, okay? And so if I observe a particular value here, I can't actually tell whether that means that there's a small reward that's more imminent or a large reward that's more delayed, okay? There's ambiguity there. So how might we kind of resolve that ambiguity? Well, it turns out that temporal difference learning also loses information about reward amounts, because what the value function converges to is the average expectation of future reward, okay? One moment of the distribution over magnitudes the system might have experienced. Now, recently in the machine learning literature, researchers in the context of kind of deep reinforcement learning algorithms found that a slight tweak on temporal difference learning algorithms allowed the system to learn information not just about the average of the distribution over magnitudes, but to learn the whole distribution. And that slight tweak just goes as follows. Instead of learning one value function, okay, you learn a set of value functions that are updated by kind of biased prediction errors, okay? And the way they're biased is that you have some channels that are more sensitive to positive errors and prediction errors and less sensitive to negative errors and prediction errors. And you have other channels that are more sensitive to negative prediction errors, less sensitive to positive prediction errors. And what happens across those different channels is that they will then converge to different statistics of the full distribution. Okay? And so this was termed distributional reinforcement learning. It was found that it produced benefits in terms of convergence times for deep reinforcement learning algorithms on a number of benchmark tasks. And then those researchers went and collaborated with Naochita's lab and found evidence that midbrain dopamine neurons, okay, show evidence of this kind of distributional coding for reward. And so you could look at the response to rewards and actually decode across the population information about the distribution. Okay. Now, this is, I just want to point out here, this is decoding information about the distribution of rewards after the fact. Okay, this is from the reward response. This is not decoding from the Q response, and we'll come back to that in a second. So we reasoned that you could actually take a kind of similar approach to the one that was used in distributional magnitude coding to extract information about distributions in time. Okay? And the basic principle there is the same. Instead of learning one value function, you're learning a bank, but in this case, they differ with respect to the discount factor that they're using to compute their prediction error. Okay? So if you do that, then if you were to look at the value reported by these different channels at the time of a queue that predicts rewards, say at differing delays, what you would see is that for a queue that predicts reward at a short delay, you would get a particular pattern of activation. Okay, it's going to have a particular shape for an early delay, and then for a later delayed reward, you would have a really different shape. Okay, so essentially, by taking almost just the spatial derivative of the response across this population, you can actually decode information about the distribution of future rewards. Right? So we set out to test whether there was evidence that the brain might be doing something like this. So we developed a simple odor-based classical conditioning task where there are 4 different odors that predict rewards at 4 different delays: immediate, 1.5 seconds, 3 or 6 seconds. And we recorded from photo-identified dopamine neurons in DAC-Cree mice. And then what we could do is we could look at the response of individual dopamine neurons to these cues that predicted reward at differing delays. And you can see an example neuron here, and this neuron, you know, its response falls off as a function of the delay to reward that's communicated by those different odors. So that's one neuron. That's the population average below. And to look at distributional coding in magnitude, we had another cue which actually predicted a reward at a fixed delay, which is 3 seconds, but now drawn from a slightly bimodal distribution of reward magnitudes, okay, to offer the possibility of looking at distributional coding along both of these dimensions. Okay? And so here you can see an example neuron in its response to reward. You can estimate the kind of slope of the response on either side of the zero crossing to derive its sort of contribution to a distributional code for magnitude. Okay, so the prediction of the theory is you should get a variety of discounting across different dopamine neurons. This is now aggregate data across 43 dopamine neurons, and indeed you can see we observe significant variability in the degree to which neurons discounted delayed reward. This variability wasn't just noise. If you compute the discount factor for individual neurons in half the trials, plotted against that computed for another half of trials, highly correlated. Okay, so indeed, lots of variability in discounting within the dopaminergic system. And this allows us to then decode information about the distribution of reward times. So this is decoding from a population of 43 neurons, just 400 milliseconds of the cue response for the immediate reward condition, 1.5 seconds, 3 seconds, 6 seconds. Okay. Now we wondered, are we just kind of as researchers taking advantage of endogenous variability that exists in the system, or is there evidence that the animal's actually using this information to guide their behavior? So we conditioned trials on whether the animal began licking early or late, assuming that's a kind of behavioral covariate of their expected time to the reward, and then decoded on those two sets of trials. And when we did that, the shift in the distribution was in the same sort of direction as the behavior in all cases. So there's at least some evidence that whatever temporal information is encoded in these responses reflects the same information the animal is using to make decisions about when to begin licking. And we wondered whether, unlike that previous study, we could decode this information about distribution over magnitudes, not just at the reward, but at the cue. And they didn't look in the cue response in that study, and we think we have one explanation as to why, because I just told you there's a lot of variability across dopamine neurons with respect to their temporal discounting, which they were not measuring. So that would have just added noise, to their data, all right? However, because we measured it, okay, we could correct for it. And when we did that, we found that the kind of distributional signal that we extract from the reward response is present at the time of the cue, okay? And that allows us to now decode information about the distribution of reward magnitudes, not just from the reward response, but also from the cue response, okay? So we have distributional information in the phasic response of dopamine populations about both distribution of rewards in time and distribution of magnitudes, and that allows us to actually combine that information and decode a map. Dean said on the first day, we know much less about how the brain constructs kind of temporal maps, where this is a temporal map, okay? It's a temporal map, the distribution, the joint distribution of rewards in time and magnitude. So this is decoding from the cue response to the certain 3-second delay. This is the variable 3-second delay, variable magnitude. And here are all the other cues. So you can see we can really read out, really just from 400 milliseconds of spiking of a population of 40 some odd dopamine neurons, this kind of model of the world, this map of the distribution of future rewards. Okay, so I'm going to finish up now because I think I'm out of time. But I just want to point out, we don't know at this point how this information is used, how it's read out. We are able to decode this information, but I have actually showed you any evidence that the brain is sort of explicitly reading out this information in this form. In fact, we know that dopamine neurons, though they project very densely and over large regions in the striatum, they do so to particular regions, okay? So there's some possibility that different dopamine neurons may send slightly different signals to different regions, and this code may actually be distributed. And recently published data, as well as data that we have in our own lab, has actually shown, and this is not unrelated to what Ugo mentioned in terms of a variety of timescales setting up in cortex, okay, that the dopamine signal seems to have differing timescales depending on what region of the striatum you're looking at. Okay, so this is now just the autocorrelation of the dopamine signal in ventral striatum, dorsal medial, dorsal lateral. And it seems that there's a sort of hierarchy of timescales with respect to the dopamine system in the striatum, which, qualitatively speaking, matches that hierarchy of timescales that you see in cortex. Okay, so I just want to wrap up by saying, my most general high-level message is that, control is layered, it's multi-scale, it has this sort of heterarchical and hierarchical quality, and so, I think it's important to point out or to think about, for those of us interested in sort of temporal processing, what is the role of that temporal processing in the particular circuits where you're studying it? What is the superordinate function of those circuits, and how is the temporal processing you see in those circuits kind of well suited for that particular function? And with that, I'd like to thank the people who actually did the work. All of the temperature work was done by a three-headed beast of Tiago Monteiro, Margarida Pichida, and Felipe Rodrigues. The dopamine work was spearheaded by a really talented graduate student who's about to graduate, Margarida Souza, who's an applied mathematician, and the experiments were done by Paweł Bujalski, who's not in this picture. Thank you.